All right, in this video for chapter 3, we're going to be discussing the prokaryotes, especially bacteria. So when we think about bacteria, we're going to think about them having a cell wall structure. The cell wall structure will be outside, so there'll be a cell membrane, and then on the outer layer, there will be a cell wall. For most bacteria, that bacterial cell wall will be composed of what is known as peptidoglycan. And we will differentiate bacteria whether or not they are gram-positive or gram-negative. So to start off with gram-positive bacteria, gram-positive bacteria have a very thick layer of peptidoglycan in comparison to gram-negative. One thing about gram-positive bacteria is that it contains teichoic acid, and that is going to help to aid in the passage of ions. You will want to know that gram-positive bacteria contain teichoic acid. They also may have lipoteichoic acid as well, and that helps to anchor the cell wall to the actual cell membrane. Remember, the cell wall is outermost, and then inside would be that cell membrane. So that lipoteichoic acid is going to help to anchor that. When we perform a gram stain, which we'll do in lab, gram-positive bacteria will appear like a purplish color. In comparison, gram-negative will be a reddish pink color. And up to 60% of mycolic acid in acid-fast genera actually help to survive desiccation. So we're going to be discussing that a little bit more in detail later on. All right, so when we think about peptidoglycan, again, this is what we're going to find in bacterial cell walls. And what we'll see is that it is basically a polymer of repeating subunits called NAM and NAG. And they basically act as what we call a chainmail armor. So it completely encloses the cell wall, and it helps to protect the cell from osmotic forces. And so when we discuss the idea of peptidoglycan, we think about these cross-linked chains and these repeating units are alternating subunits of NAM and NAG. And so again, when we think about this cross-linking, this is going to be the structure that we see. So very important to think about them as cross-linking, and again, think about it being that chain mail armor. I'm not going to ask you to know the detail, but again, just to think about sort of what NAM and NAG are. They are derivatives of the glucose molecule, and as you'll see, they have a little bit of a difference in them. So when we look at NAG, it stands for N-acetylglucosamine, and if we look at NAM, it's going to represent N-acetylmuramic acid. Again, you don't have to know these in detail. This is more just to see that it is a derivative of glucose. And again, when we start to look at that gram-positive bacteria, as we may mention, there's that very thick peptidoglycan cell wall, and that's going to be represented in that purplish color. We'll also see that teichoic acid and lipoteichoic acid as well. Remember, the function with lipoteichoic is really to actually anchor the cell to the cell membrane. And again, you'll be able to see how there's that cell wall, and then underneath we'll find that cytoplasmic membrane or cell membrane. And this is going to be a nice little picture to be able to see those differences. I would suggest knowing this picture and what you are seeing. So again, understand that phospholipid bilayer that we're seeing, understand that this NAM and NAG repeating subunits, the teichoic acid and lipoteichoic acid. So I would greatly suggest knowing this figure. All right, next we're going to discuss gram-negative bacteria. Gram-negative bacteria will have 3 layers. There will be an inner, or what we call a cell cytoplasmic membrane. Then we will see the peptidoglycan cell wall, and then there will be an outer membrane on top of that. So think of it as being 3 distinct layers. Again, the inside layer would be that cytoplasmic membrane. Then in the middle, we'd find that peptidoglycan, and then an outer membrane. When we think of the peptidoglycan cell wall, it's going to be a lot thinner in comparison to that gram-positive bacteria. So we say that it has just one very thin layer of peptidoglycan. On the outer membrane, though, what we're going to see is that it's going to contain phospholipids, proteins, and in particular with gram-negative bacteria, you should know of LPS, lipopolysaccharides. These lipopolysaccharides are going to be very important when we start to discuss endotoxins later on, talking about disease presentations. Gram-negative bacteria will have this lipopolysaccharide that can actually break down and release an endotoxin when the cell is damaged. So that's a very important part of gram-negative bacteria. What we'll also see in that outer membrane is that there's going to be porins as well. And we're going to see that there's a space in between called a periplasmic space. Now, when we look at gram-negative bacteria, they will stain like a pinkish red color, again, in comparison to gram-positive bacteria that are going to stain a purple color. So I always remember positive purple and then negative red. So again, more like a reddish pink color. All right, and this is going to be able to show you a little bit more of that difference. So again, on that outermost membrane, we find those porins. We're also going to see again that we have an outer membrane. In between the cell wall and the cell membranes, we're going to notice that there is a periplasmic space. Again, we find one thin layer of peptidoglycan for that cell wall, and then we have this inner membrane. Now, keep in mind as well on that outermost membrane, we're going to find that lipopolysaccharide or LPS as well. And again, this can be released if the cell becomes damaged and can create an endotoxin. All right, so again, another picture I would greatly suggest knowing so that way you can be able to identify it and label it. And this just allows you to see that difference between gram-positive and gram-negative bacteria. Remember, gram-positive bacteria, we're going to find that they have that lipoteichoic and teichoic acid. Again, we will find those repeating subunits or alternating units of NAM and NAG. We're going to see that there's a very thick layer of peptidoglycan, and then we'll find that cytoplasmic membrane or cell membrane. With gram-negative bacteria, we'll find three distinct layers. So again, we have a cell membrane, we have a thin peptidoglycan cell wall, and then we have this outer membrane. In the outer membrane, we're going to find porins, we're going to find lipopolysaccharides, as well as we will also find, again, repeating subunits of NAM and NAG in that cell wall. All right, so what differences does it make if a bacteria is gram-positive or gram-negative, you might ask? Why is it important that we actually stain for this? And really, why do you care? The big thing is antibiotic treatment. The way that we would go about basically creating a defense mechanism against this bacteria would have to do with the antibiotic treatment. So when we think about penicillin, for instance, penicillin is very useful for gram-positive bacteria. And penicillin derivatives disrupt the formation of peptidoglycan. So if we have a faulty peptidoglycan, then we're not going to have a great cell wall structure. And because they won't have that proper cell wall structure, eventually the cell will rupture or what we call lyse, and it's going to die. So the outer membrane of gram-negative is going to actually protect the cell from penicillin. So while this is really helpful for gram-positive bacteria, this isn't that wonderful for gram-negative bacteria. And so this wouldn't be the drug of choice if you had a gram-negative species. Now, not all cells actually have cell walls. For instance, mycoplasma does not. So you'll hear about mycoplasma pneumoniae in particular. And so it doesn't have that same peptidoglycan cell wall. So therefore, we wouldn't actually run a gram stain to be able to identify this particular organism. All right, now thinking about other structures we could potentially see, some microorganisms will have a capsule. So when we think about bacteria, we can talk about them having a glycocalyx or capsule. It's made up of polysaccharide fibers, and it's basically a layer of this mucoid material that's going to surround the cell. Sometimes we discuss it as a capsule or a slime layer, but the ultimate functions are going to be that it helps to actually protect the cell from drying out, so it prevents desiccation. It's also going to help bacterial cells from phagocytosis. And what phagocytosis does is it engulfs that bacteria. So our white blood cells in particular, macrophages, something called dendritic cells, they're going to be able to grab the bacteria, basically engulf it, and then break it down. That capsule or that glycocalyx actually protects the bacteria. So while it's great for them, it's not so great for us. And it also helps bacteria to adhere to cell surfaces, such as what we're going to see with like plaque on our teeth. It helps to actually create what we know as a biofilm. All right, some other structures that we'll see externally are flagella. These are going to be tails that are used for motility. And unfortunately, they're too thin to be seen with a light microscope, so we would have to do a special flagella stain in order to see them. Some microorganisms, or I should say some bacteria, may have one flagella. They may have one on each side. They may have some coming out of both ends, or they may see it surrounding. If you see it surrounding the entire organism, this is called a peritrichous flagella. Trich, think hair, and peri, surrounding. If you just see it on one side, it's going to be called a lophotrichous. You may just see one, which would be called a monotrichous, or you can see them coming out of both ends, which we would call an amphitrichous. You may also see these pillae or fimbriae. Fimbriae are going to be a lot shorter and thinner, and we tend to see them more numerous than what you would see with the flagella. So you'll see a lot more of these fimbriae. You also may see this pillae as well. And their ultimate goal is to enable attachment to surfaces, especially to their host. Now, with pili in particular, they can be a sex pilus, and it helps to transfer genetic material, like, for instance, what we'd see as a plasmid. So remember, the plasmid is that additional chromosome or that additional genetic material that a bacteria may have. And not all bacteria will actually have a pili, but again, it's helpful in what we know as conjugation. We'll talk about conjugation coming up in a few chapters from now. All right, and this just allows you to see that conjugation happening. So if they're able to form this sort of sex pilus and form this cross bridging, if you will, this allows them to swap that genetic material. So be able to have a donor bring that plasmid to a recipient, or at least copy it and send it over to the recipient. All right, in terms of bacteria, these little additional or extra cell structures we may see, some bacteria will form what are known as endospores, and some bacteria can form what are called biofilms. So endospores are a major problem, and they are wonderful for some bacteria. They are very, very difficult for us to be able to treat. And what endospores are dormant structures that are going to allow for resistance of the bacteria. They're not found in every bacteria, but they're found primarily in gram-positive bacteria. And cells that actually produce endospores don't always have the endospores. Normally, the cell is going to exist in a vegetative cell or as a vegetative cell, which means it's actively growing and reproducing. However, if conditions are not favorable, so maybe there are a lack of nutrients to prevent the cell from drying out or desiccation, or it's been exposed to high heats or changes in pH, this vegetative cell, this normally growing cell, can undergo what's called sporulation. And through sporulation, there's a series of events. So usually when we talk about this vegetative cell undergoing sporulation, usually it's in response to issues with nutrients, maybe there's high cell density or other environmental factors. And usually they have no detectable metabolism once the endospore has been formed. So now they're going to go into a dormant stage. Kind of think about it like a sleeping beauty mode, if you will. Now, when they are exposed to favorable conditions again, now they can actually germinate and go through this cycle in order to become an actively growing cell once again. So this would be what they consider to be germination taking place. And so this just sort of shows you this idea of the spore cycle. So again, if it is a vegetative cell, it's actively growing. But again, let's just say hypothetically, it's been exposed to very high heat or radiation, then it can form this endospore. Now, again, the endospore for time frame that this happens in is about 8 hours. And it can be an endospore for a long time. It can be in this dormant stage for years. But if something happens where it's now exposed to a favorable condition, now it can germinate. And so the figure that you can see on the side actually kind of shows you that cycle of when it goes through that vegetative cycle versus that sporulation cycle. You won't be asked to know every detail to this endospore cycle, but you have to know that it can be a vegetative cell, undergo sporulation, and then become an endospore. And like I said, endospores can live for a very long time. They can be in this dormant stage for years and even centuries. And the big problem that we deal with endospores is that they are resistant to extreme environments. So they can withstand very high heat. And because of that, in order to eradicate them, we have to sterilize using an autoclave. They can be exposed to chemicals, they can be exposed to drying out, so desiccation, high volumes of radiation. And so, because of that, we have to sterilize in order to remove endospores. Now, one thing that they are not is resistant against antibiotics. So that's just one thing to make mention of. But again, heat, chemicals, drying out, high UV radiation, those are all things that they can be resistant to. So we have to use sterilization techniques in order to be free of endospores. And then we also can deal with biofilms. So biofilms are a community of a variety of organisms that will attach to a solid surface, and they usually will grow in masses. So we'll see them on rocks, on teeth. So when you think about like dental plaque or dental caries, and they can unfortunately grow on medical equipment as well, primarily implants. So this is a big issue where again we want to make sure that we have a proper sterilized implant, for instance, like a prosthetic knee or a prosthetic shoulder, so that way, when it is implanted into someone, it's not containing this biofilm, this variety of microorganisms that are prokaryotic and eukaryotic that can eventually cause disease. All right, so when we see this picture here, this is going to be looking at biofilms for a catheter. So again, this should be a catheter in all reality should have been sterilized and free of any life forms, but we can see this biofilm formation with the Staphylococcus species taking place. All right, couple other things to make mention of. We want to talk about the cells in terms of morphology and arrangement. When we think about this microscopic analysis, so what we're seeing under the microscope, we're going to see shapes. So we're going to discuss morphology. And there are three basic shapes that we should see. A cocci is going to be a spherical shape. A bacilli is going to be a rod shape, and we can also see some spirals such as spirillum or spirochetes. The spirillum are going to be like a helical shape or a spiral shape, so think like a slinky. But we can also see some other shapes, like a vibrio is going to be like more comma shaped. The cocobacillus is going to be a more blunt rod. And then we can also see pleomorphic, which would mean that they can take multiple different arrangements. And that's what we can kind of see going on in this picture here. All right, now this gives you a little bit more detail to see that arrangement. So again, we can see them as pairs. So we can see them as a diplococci. For the cocci, we can also see them in chains, which we would call streptococcus. We can see tetrads, which would be a group of four. We can see the sarcemia, which they're going to be more cube-like. And then finally, we can see the staphylococcus. And staphylococcus would tell us that they have like more of a grape-like cluster or an irregular cluster taking place. And same thing for bacilli. We can see one bacilli. We can see it in pairs. So we'll see those two rods. Strepto would tell us that the bacilli are in a chain. And we can also see this palisade or this V shape taking place. All right. Thank you so much. And I hope you have a wonderful day.